Hi everyone, you're welcome to once again to our Quantum Jollof podcast. Then for today we have our guest, and the name of our guest is Dr. Oyebola. And I'm going to tell you a little bit of some of his background, and then we can move to the program for today. Dr. Oyebola is a data scientist, a bioinformatician, a geneticist, and cybersecurity analyst, and he specializes on genomic research, AI, machine learning. Dr. Oyebola had his postdoctoral training at the London School of Hygiene and also the National Institute of Health in the US. He has led several research projects that was funded by several global institutions, and his work has been an advancement of genomic data analysis for malaria, sickle cell disease, and he has used big data and cloud computing, machine learning, and other platforms. He serves as expert on different boards, he mentors scientists, and he runs a research lab and he has PhD student that he supervises. Additionally, he specializes in cybersecurity, forensic investigation, data privacy, and most importantly, he founded the Center for Genomic Research in Biomedicine at Mountaintop University and also the Telecare Medical Couriers. Dr. Yoboda, you're welcome to our Quantum Jollof podcast today, and that's a lot of achievement.
Yeah.
I'm definitely inspired by this. I hope that one day someone will be able to read my, citation like this and they will,
Yeah.
and I when I'm reading through this and I'm actually smiling because this is a lot. So
Well, thank you for hosting me. I really appreciate this platform. I am also very excited about what you're doing about Quantum Jollof. Uh, your passion about science, and of course, trying to encourage others to learn more about science, especially the younger ones, to understand the details of, and of course, to kind of carve a career path in science, which is really encouraging, and I'm really excited to be here.
thank you. So Dr. Iwola, we have a lot of questions for you today, and we hope that you answer most of our questions, our silly questions, sometimes silly, sometimes serious, but we'll try to keep it real. And I'll open the ground for Wale to go with his first question
Okay. So
today.
yeah, can you describe your scientific journey so far, and where did you start from, and how did you end up researching sickle cell malaria and sickle cell disease and malaria, two of the toughest health challenges I would say in the tropical world?
Yeah, thank you very much. Um, it's a long story, like we always say, right?
Yeah.
But then I, when I was introduced to genetics as an undergraduate, I really enjoyed genetics. I I realized that I could explore that field, in different organisms, animals, plants, humans. So after my undergraduate studies, I knew that I was going to advance and in the field of genetics to learn more about genetics. And then, of course, it was at that time that um, bioinformatics, came on board, analysis of genetic data mostly. So I I realized that, of course, if I have to really do laboratory genetic analysis, I would require computational analysis to be able to get to the end point of my project, my research work, meaning that I needed to learn bioinformatics. So I did my PhD in bioinformatics, parasitology and bioinformatics at the University of Lagos. And just about the time I was rounding off, actually, I got the overall best PhD thesis award during my set, during my time. And just about the time I was rounding off, there was this opportunity to compete or applied for a Wellcome Trust funding that was localized in Africa for recent PhD graduate. Of course, I was still, I just submitted my thesis, so I was waiting for the examination, the final thesis viva. So I spoke with my supervisor then, who was then at the Medical Research Council in the Gambia, affiliated with the London School of Hygiene and Tropical Medicine. And he encouraged me to apply, and it was really encouraging. Professor Alfred Amambwa Ongwa, very supportive, and I spent some time, days and nights trying to get the proposal ready. And just about the time I was, called for my thesis examination, I was also invited for the interview to be awarded the fellowship, and the fellowship amount was 150,000 US dollars. So I spent 3 years at the MRC at the London School of Hygiene and Tropical Medicine, working on malaria genetics. And just about the time I was rounding up, there was also another opportunity to apply my genetic skills, my knowledge in genetics. So in this case, to apply it in humans at the National Institute of Health in the US. So I got the award. That was like a 5-year research fellowship that took me to the US, where I started working on sickle cell disease with my mentor at the NIH, and I found it really interesting because, of course, there is an intersection, more like a meeting point between malaria research and sickle cell genetics. If you look at the evolutionary arms race between malaria and sickle cell, you realize that um, malaria kind of, gave selective advantage, or more like um, in the mutation of the hemoglobin gene, that's usually the incriminated for in sickle cell disease,
Yeah, I
because
think we will talk about this anyway. So yeah.
let me just land
Okay.
on this point.
Okay.
So the mostly Africans and people in sub-Saharan Africa and maybe some parts of Asia are exposed to malaria, meaning that to protect against malaria, there was some sort of evolution in that gene, which gave selective advantage to individuals who had a mutation in the gene. Um, so I'm just trying to explain the relationship between malaria and sickle cell disease, which I'll talk about much later. So that led me to the US, where I, gathered more knowledge about not only genetics and bioinformatics. Then I became exposed to data analysis. How do I really analyze high throughput data if I don't want to really go through this, phase of wet lab analysis? I want to use already data that is available in the public domain. I have thousands of data sets to analyze. How do I do that? So I realized that machine learning and AI could help me. So I had to learn. I went back to school to get a diploma in data science and AI, and that led me to AI analysis. And then from there, I got research grants. I won research grants and all awards to set up a research lab in Nigeria. And because that was my midterm goal, really, to set up a research lab that could staff for capacity development and train the next generation of scientists. And with that, I set up the lab, and then of course we've trained um, a number of students who have gone ahead to do something, or other um, uh, significant things. Um, and then of course I've been applying my science skills in different fields, being involved in different consortia. I've set up businesses, science-related businesses, um, published, papers, manuscripts, and books. I mean, I'm just doing my science the way I know how to do it best. I just want to make a mark, in the science world, to touch the lives of the next generation of scientists, and of course, make you know life and people healthy. Discover new drugs, or maybe vaccine candidates for malaria, and of course also explore sickle cell disease research to see how, because these are two diseases that plague, actually they scourge Africans a lot, malaria and sickle cell disease, and most likely if they're not global diseases, they tend to be neglected by um, the rest of the world. But we have to look for solutions for these diseases ourselves, and then I have taken it upon myself to research the diseases, and then of course see how far I can go in looking for solutions to them.
Yeah. That's quite a story. Like, I'm inspired, honestly. Like, I had to go back to your LinkedIn to like see again. Like, yeah. Oh, I have a question before I move on to the next question. Like, in your journey right now, do you still like run a wet lab, or that part is carved out?
So I do sometimes, just that I have staff that do most of the lab analysis for me. What I do mostly these days, I do a lot of writing. Um, just like I told you, I have three submission deadlines tomorrow. Um, One of them is like a manuscript deadline that I have to review as part of a bigger collaboration with other groups that we're submitting to Lancet. Um, and then, of course, two of them are research grant applications that I have to submit tomorrow. Just a few days ago, I submitted another research grant application. So it's more like what I do mostly now. I do data analysis also for most of my manuscripts. Um, and of course, maybe if, because I also have staff who work on data analysis, but if they run into any brick wall, or they need to troubleshoot anything, I have to also get involved in that. So that does it. I stay in front of my desk most of the time, right?
That makes sense. So, moving back to like sickle cell disease, like can you tell us about sickle cell disease? And I know a lot of people a lot of family that have been impacted by this disease, and this is a big problem in Africa, especially. So, why is this difficult? Why is this disease so difficult to cure all of these years? I know there's a lot of research, a lot of programs have people have put out there to like tackle sickle cell disease, but why is it so like difficult to like to tackle?
So first of all, there is a cure, right?
Mm.
If you have been following the news, you realize that information came out of the UK that that was a cure. Of course, it's not new for some of us in the field. We were not really surprised, or we didn't think that was news to us, because we had been following the trend. And as a matter of fact, at the NIH, a lot of work had been done on sickle cell disease cure. Research looking for cures to permanently fix sickle cell disease. First, we need to understand the disease, right? It's a blood disorder, and it's hereditary, right? Meaning that it can be passed from parent to offspring, and it's caused by a point mutation in the hemoglobin gene, HBB gene, located on chromosome 11. So, and the mutation leads to um, the sickling of the red blood cell, right? The red blood cell is supposed to be flexible, so it can pass through the vessels, right? But when it becomes sickled, as a result of there'll be a lot of sticking together, polymerization, and then of course it blocks the vessel, right? That blood passes through, meaning that there's vessel occlusion, and of course, when there's vessel occlusion, limited blood passes through the vessel, and limited amount of oxygen gets to the organs, right? So you can imagine, you need maybe 100% oxygen at and then of course it goes down to 20, meaning that of course the organs will not be operating at the optimum level. So vessel occlusion, like I mentioned, is a very common phenomenon in sickle cell disease, and of course the red blood cells also lyse. Most times the lyse, they are very fragile. They lyse quickly, they break apart quickly, leading to chronic anemia, right? Of course, that means there's, there are fewer red blood cells in in circulation. So the end result could be organ damage, because oxygen has been, the organs have been deprived of oxygen, kidneys, lungs, livers, um, spleen, even to the brain, because when oxygen doesn't get to the brain, it can cause stroke, or there's also that tendency for pulmonary hypertension and kidney failure. So it's a very, the end result for it's painful. It's a very painful disease that, it's when you experience someone struggling through the phase or the crisis, you feel really bad for them, because it's very painful. Every the bones become painful. There's almost damage to every organ of the body. So it's not a very. I feel, when you're doing this kind of research, you're doing it with a lot of compassion, because you can imagine the pain people feel as a result of just a point mutation in the HPV gene. I don't know if that answers the question.
Yeah,
Yeah, that was very, very deep. And yeah, sickle cell disease, I know some friends back when we were in secondary school. It's it's a very big issue. It's a very big issue. Um, so so one question. So you were talking about like some of the long-term consequences of sickle cell disease. And in some of your previous studies, I think the one you did at NIH, we did some background research anyways. So we realized that you studied clonal hematopoiesis in sickle cell patients. And it somehow leads to maybe some form of cancer. Is it that sickle cell disease can lead to cancers as well?
Okay, so thank you very very much for the question. Very brilliant question. Um, so first we need to be able to define the the phenomena that you've mentioned. You mentioned clonal hematopoiesis, you
Mhm,
mentioned cancer, you mentioned also sickle cell disease, and how do they now relate? First, we need to understand what hematopoiesis is, right?
mhm,
The production of blood from the stem cells, right?
mhm,
So, as and mostly it occurs in the bone marrows. So, when you have blood cells produced and there's a mutation in any of the, so let's say we have 10 blood cells, right? Stem cells. And one of them has a mutation, a somatic mutation that is, that predisposes in people to blood cancer, to cardiovascular diseases, right? So we have 1 out of 10 cells, right, with
mm,
that mutation. And imagine in sickle cell disease, just like I mentioned earlier, right,
mm,
that the hemolysis takes place more often in sickle cell disease patients than in normal living individuals. So imagine the other 10 or maybe four of the other cells that were produced have lysed, leaving the one with the mutation. there will be clonal expansion, meaning that the one with the mutation will continue to multiply and multiply and multiply.
Mm-hmm.
That is, so it already has a selective advantage or a fitness advantage, right? So in normal aging individuals, we can only have that maybe one mutation out of 10. After some time, because there are 9 other good cells, the one mutation probably will take a lot of time for it to expand. But in sickle cell disease patients, it takes lesser amount of time, meaning that the uh, the consequences or sequelae of having that mutation, manifests earlier in sickle cell disease patients. So the somatic mutation associated or that predisposes people to say blood cancer or cardiovascular disease, it will manifest earlier, right? Sickle cell disease patients than in normal aging individuals. And secondly, because there is also inflammation. Sickle cell disease is a disease of inflammation. It's associated with inflammation. The production of cytokines and chemokines is, accelerated. So as a result of this production of these cytokines, the the clonal cells also expand. They increase in population compared to the normal cells. So inflammation, hemolysis, uh, bone marrow hyperactivation, because of course when blood cells lies, more and more cells will be produced, meaning that there's that rapid increase in the number of cells produced, which may also be the cells with the clonal mutations. So all of this combined together will now make sickle cell disease patients more prone to somatic mutation related diseases or crises. It could be blood cancer, it could be cardiovascular diseases. And of course, now added to the already, all the physiological processes going on leading to endocrine damage and all that. So that's why it's important to really study if these mutations are present, right? So that we can monitor the progress or expansion of these mutations, or even start treating them, because many of these mutations can be targeted by drugs, so that the endocrine damage or the consequences of accelerated expansion of these clonal cells will be minimized. So that's the essence of studying clonal hematopoiesis in sickle cell disease patients, right? I mean, I also studied this in normal aging individuals because, of course, this somatic acquisition of these somatic mutations really increases with age as you grow older. Of course, the clonal cells begin to multiply, and that's why you've realized that older individuals are more prone to cancer, more prone to cardiovascular diseases, right? And all sort of inflammation-related diseases. So that's the essence of studying this in sickle cell disease patients.
Dr. P, I actually want to take you, I am an African man and we believe a lot in assumptions and superstition. So I want to probably take you away from science a bit, but I'm not sure you are not even going to take me back to science. But the next question I have for you is that as Africans or as people, we have this superstition that we believe that if someone is carrying like a sickle cell gene or sickle cell variant, they are less susceptible to malaria infection. And I can say that I've practically seen at least one or two people from my environment that might testify to this, but there's no scientific backing to it based on the African mindset. So how true is this? Can you shed more light on this?
So actually, thank you very much. That's also another important question. There is scientific back. It's not a myth. It's a fact, right? There's scientific backing for to show that people with not sickle cell, the sickle cell trait, the sickle cell trait itself, the sickle cell gene, they are not like they won't be infected. They are not immune to infection.
Okay.
They will still be infected. Just that their red blood cells are less hospitable to malaria parasites. And if you go back to what I've explained, or I will explain again, but if you go back to what I've explained, you realize that you realize why this is the case. Now, imagine you have your normal red blood cell, which is flexible, and then of course you now have a sickled red blood cell, which most likely polymerizes, clusters, and hemolyzes quick. And parasites are in the sickled red blood cell. Don't forget I mentioned that hemolysis takes place much faster in sickled cells than in normal red blood cells, right? So of course, by the time hemolysis takes place in those cells, the parasites will just be released and then they die. They get exposed to the immune system, and then they die, right? So it takes away most of the complications associated with malaria. They can still get down or be infected with malaria, right? Malaria parasites, but the complications, um, malaria-related complications like severe malaria, cerebral malaria, or death as a result of malaria is reduced. They are reduced, right? Those
Mm.
complications are reduced in individuals carrying the trait, right?
Mm.
So protection is not absolute. They're not immune to infection, but of course, they have a lower risk of severe forms of cerebral malaria or complicated malaria. Does it make sense?
Yeah, that
Yes.
makes sense.
Before
And
you to the next question, please, can you quickly touch on blood genotype? Because so we are talking about sickle cell, the sickled one, and I know there is an intermediate between the absolutely round one and the sickled one. So can you just touch on those blood genotypes just for clarity?
Okay, so for the genotypes, of course, the genotype is defined based on the genetic makeup of the hemoglobin, right?
Mm.
The oxygen carrying protein in our red blood cells. So the presence or absence of those sickle cell um, hemoglobins, right, defines your genotype, right? So if you, for instance, have your genotype is AA, that means you have the normal hemoglobin, the HbA,
Mhm.
right? You're healthy, you have no risk of sickle cell disease.
Mm.
And however, if you have the sickle cell trait, like one of the hemoglobins, we have the one normal gene, and you have another one that is sickle, right? So one that is AS, right? That's the sickle cell trait. That means you're a carrier. Sickle cell carriers are mostly asymptomatic,
Mm.
meaning they may not have symptoms, but or sometimes they have mild symptoms
Mm.
in extreme conditions. Maybe they when there's dehydration, or maybe they're at high altitude, they they start feeling that sense of the what the pain, just mild pains that the sickle cell disease, homozygous sickle cell disease patients feel. But it's usually mild and asymptomatic. In rare cases, there could also be, they could be, they could experience crisis, right?
Mm.
Um, so we have the A, we have the AS, right? Of course, and then of course we have the SC, which is the minor form of the sickle cell disease. Actually, almost the full blown sickle cell disease patients,
Mm.
right? It's milder than the SS patients, but it's more complicated than AS, right? So, but there could be complications associated with SC. Then, of course, we have the SS, right? The
Mm.
severe sickling, which most likely leads to anemia, pain crises, organ damage, stroke, accelerated aging, um, you know, name it, all sorts of complications. Of course, susceptibility to even communicable diseases, right? And we have also the S beta thalassemia, which is the sickle beta thalassemia, which varies in severity depending on the beta thalassemia mutation, whether it's mild to severe. So, but our major focus mostly will be on, if we're studying sickle cell disease, will be on SS and SS.
So because of your time, I will just like move on to the next question. I know you mentioned that there's currently a cure for sickle cell, which I actually didn't know. I have to be honest. But is it that this cure is not readily available to people from less or developing countries right now, or what? And how efficacious is this cure? Like, is it really efficacious, or is still like under development?
Yeah, I mean, it's there are several factors. Thank you also for the question. There are several factors involved here. We have the economic factor, we have also the research factor, and maybe the genetic factor. But before I go into the details of those factors, maybe limiting the accessibility or availability of the cure for sickle cell disease, let me just go into some of the available um, um, therapies, or treatments, potential treatments for sickle cell disease. Right? So maybe we'll go back to the sickle cell, just like I said, it's a single mutation, point mutation, glutamic acid to valine change, substitution at position 6 of the beta globin, right? So on that basis, we know where to target. And so gene therapy targets this region, right? It's kind of straightforward. It looks like a straightforward solution, right? However, as a result of genetic complexity, which challenges with the delivery of gene therapy, now that we have CRISPR gene editing tool, it makes things easier. Even though it's technically demanding, but of course, that used to be a challenge. Delivering the gene therapy was a major challenge in the past, but of course, CRISPR now comes into play to solve this. To probably look into the solution to the challenge. So delivery used to be a challenge, and because of the genetic complexity of the sickle cell disease, and then of course the cost, right? Let me not go to the cost yet, but let me just talk about even within the gene therapy solution. There's that requirement for follow-up, right? There's that need for long-term follow-up, which in many cases, especially if you're involved in longitudinal studies, research work, humans are difficult to really follow up, track down for a long period of time. So that's also it's a challenge. So the durability of the treatment has been could be hindered as a result of that. And it's not only gene therapy. There's also another solution. If you look at the bone, just like I mentioned earlier, right? Bone marrow releases this, produces stem cells, right? Cells that are carrying the sickle cell hemoglobin. What if you target the roots that's producing those cells, wipe away the bad cells, and then introduce new cells? That's the essence of hematopoietic stem cell transplantation, right? So there are two types. You probably have the autologous, which means that you're taking cells from the same individual to replace maybe from one part of the body to replace another region in the bone marrow to start producing new fresh cells. But of course, that's not going to happen because it's the same individual. So, and the other type is the allogenic hematopoietic stem cell transplantation, which involves donors, maybe close family members, siblings, who can donate their blood. So there are complications associated with this, and I'll mention a few. First, getting the donor that completely matches whose HLA. HLA is human leukocyte antigen that matches with the recipient. It's usually very difficult to get a match. Even at that, even when you get a match, there's that possibility of a rejection. Yeah, the immunity of the host may reject the the and the transplants. I mentioned two complications resulting from that. There's also a third one, which is there are some times where, because you have to completely wipe off the stem cells from the bone marrow, the original stem cells, there are some times where you cannot achieve that. Some cells are left, and these cells could be the ones carrying clonal mutations, meaning that they will be probably more exposed to or more prone to somatic mutation related diseases, complications like the ones I mentioned earlier, like blood cancer, cardiovascular diseases, right?
Mm.
There are so many other, complications. I don't want to really take some of the time, but it's also a very risky procedure, right? In the process of transplantation, there's risk of infections. I mentioned the risk of rejection and even death, right, unfortunately. So it's complicated, and then I'll go to the main factor, which may be a limiting factor in terms of the availability and accessibility of the treatment, because it is very expensive. It's usually about more than 1 million. I don't know the current rate, but of course, as of last time I got wind of the information that it could be for one patient, right? It could cost more than $1 million, right? So imagine in Africa where this actually is endemic, right?
What is
Right?
our earth budget?
So who's going to be able to afford it? And if government decides to subsidize it, government, let's not even go into the issue of government, but there isn't they don't even have the money to subsidize it. So until it becomes affordable, it cannot be said that there's a cure, there's an accessible or available cure yet, right? There are other, there are other reasons. Even if you get the cure, a lot of damage might have been done already. Just like I said, depending on the age of the individual at the point of receiving treatment, there could have been, end organ damage, the complications, related to the heart, that may not be, treatable, even if you completely get new blood cells into the patient or completely reverse the gene as a result of CRISPR-Cas9 or gene editing tool. There could be such complications, and of course, there's a way to manage it. There's the hydroxyurea. I mean, it's not all gloom and doom. There's hydroxyurea, right? That's available, which and then of course that's the mode of mechanism of action, or the mechanism of action of hydroxyurea is to increase fetal hemoglobin, right? So the BCL11A gene that in all of us, right? When we are born, we have a high level of fetal hemoglobin as infants. As we grow older, the BCL11 gene comes into play, reduces to the barest minimum the amount of fetal hemoglobin we have. So to encourage or to boost production of fetal hemoglobin, there's the hydroxyurea targets BCL11A to allow production of fetal hemoglobin even in adulthood, which helps with managing sickling, and of course reduces the pain or crisis, minimizes it. Not complete. It's not a complete cure. It's a management drug. So it's not a doom and gloom. If it can be, of course, if also the actual fix can be affordable, can be made affordable to the people who really need it, then of course we probably have a solution in sight.
Interesting. I mean, that's well, we hope that with talented scientists like you, we can maybe one day have a solution to sickle cell. I mean, I think it's a really, really big issue. And if it's left to, for example, the United Kingdoms, they will keep developing solutions that are out of our
Out of reach.
yeah.
Yeah.
Yeah. Okay, so because of our time, we would like to round up, but before we round up, we'll give you the opportunity, what we call a nuance window. So the opportunity to talk about anything that we probably missed so far, what's on your mind, your broad perspective, either about sickle cell, about malaria, about Nigeria, where you come from, any bigger lessons that you think it's important for everybody in the world to know about your research on sickle cell in general.
Yeah, I mean, I really there's one thing that bothers me about science. It's science is in danger, is in jeopardy at the moment. Um, Attention of world leaders to the importance of science, of research, as really is too is discouraging. It looks to me like they don't understand that science actually births all these drugs, vaccines, discoveries that we have in everywhere across board, in medicine, in tech, everywhere. And because of this, the global attention is fading, and we in Africa we are not alive to our responsibility, particularly the leadership of African institutions or countries. They are not alive to their responsibility. We rely so much on external funding. Some of them don't even understand why we have to start, or we need to start funding research, because the world is becoming, or each country or is becoming too nationalistic. They are becoming so focused on internal challenges and problems, solving those problems, that they don't care about what happens in other regions. And it's a wake-up call for African leaders to realize that, God forbid, if anything like COVID happens in Africa, for instance, again, there's a likelihood that Africa will be locked out to whether to die or whatever. The rest of the world does not care anymore. The leadership of the rest of the world does not care about what happens. There's no global plans. There are no global plans anymore. There are no there's no world view to research and everything. So the essence of what I'm saying is that we have to really pay attention to science and technology in Africa. Otherwise, we may be wiped out of the of the world, and wiped out, completely removed from the map of the world. Because if there's any epidemic that spreads so fast that allows the rest of the world to shut their borders, then of course we'll just be left to be wiped out. And I hope and pray that that doesn't happen to us. And one way of doing that is for for us to fund research, establish world-class institutions across different regions in Africa, train world-class scientists, and of course encourage them to remain in Africa to do their research by making um, um, doing their work in Africa uh, uh, attractive and rewarding. Otherwise, I can't imagine what will happen, because as we are now, there's no incentive for anybody to want to do science in Africa. No incentive, none at all, none.
They want people to like come back to Africa. They need to make Africa to be conducive. Like,
Yeah, there's no incentive at all for anyone to want to do science on the continent.
literally in my
No,
current
none.
our electricity went off for like 2 hours, like 1 hour, and everybody was panicking in our group chat. In my lab here, just 1 or 2 hours, electricity just went off. Now imagine Nigeria where
Because I have the experience, I'm doing a lot of heavy lifting. I feel like we can, Adewale, I feel like we can, you know, share with you some of the things we go through. Um, it is frustrating. Um, and I'm not even talking about, of course, they don't give you anything. Of course, one cover they won't give you, yeah. But in terms of the basic things needed, like electricity, like even you can even provide your own electricity, support,
right,
institutional support, moral support. These things are not there. You have to beg, kowtow. You have to sometimes even give some people money to so that you can just do your science. It is it is painful. And if you're passionate about science, are passionate about your country or your continent, you won't. There's no way you can be happy about this. It
right.
is really disgraceful.
Oh, we hope that one day touch the heart of the electors, because it's not the leaders right now. Even the electors, we are part of the problem. Both the
Yeah.
leaders, the followers. God should touch our hearts, and then maybe from there we'll be able to move on to do greater things. Hopefully. So
Uh, it's looking like a mirage. Well, who knows?
yeah,
A miracle can happen. Yeah.
happen exactly. Yeah, yeah. So yeah, Doctor, hey, it's really nice to like talk to you about your science, about your journey, about everything that you've been doing, every great things that you've been doing. I'm personally inspired, and I hope to be able to follow that kind of path and to be able to like do great things in my own field as well. And we really appreciate you for accepting to join us today. And we pray and hope that every of your endeavors will manifest, everything you pray for will manifest in the name of Jesus. Then thank you for joining us, everybody that is watching this video. And I don't know if Wally has anything to say, or otherwise we have come to the end of the podcast for today. Thank you.
I would just like to say thank you once again for joining us. It's you've always been an inspiration. Yeah, and I hope every other person watching this or listening to this would get inspired as well. Yeah, it's a really long journey from Lagos to the UK to the US, back to Lagos to Canada. Oh, wow, yeah, that's some move.
And only research lab in Nigeria.
Nigeria, yeah, that's something for legends. Yeah.
Thank you very much, and I really appreciate this platform. Just like I said earlier, keep up the good work because I know you're also touching um, lives. You're impacting the way you can. You're passing knowledge to the next generation of scientists, which is very encouraging, and I'm excited about that. I'm delighted whenever I see something like this happening, especially you know in our world in Nigeria.
Yeah, yeah,
it's uh,
yeah.
Not so many people talk about science in Nigeria, and it's very, very interesting. All right. Thank you so much. We
Let's-
hope that you are able to meet all of your deadlines.
yeah.
I hope so. I hope so. I
You
just have to
set up the boundaries.
right now, then of course start my work.
Okay. Thanks a lot. Bye. Yeah.